Good afternoon, fellow students. My name is Professor Dmitry Starchik, and we again meet together just for human anatomy lecture. That is the lecture. We start the section of central nervous system, but that is a special part of the systems devoting to the sense organ. And today we start the anatomy of the vision organ, or in other words, organ of vision. I see today 5%. That is good for now, and let me know as usual. I always ask you about the quality of the translation. Please give me your feedback. Thank you. Well, come to the educational material. Thank you. This is our today topic is organ of vision. Anyone who enjoy music, art, fine food, or a good conversation appreciate the human senses. I hope somebody of you like music, and a lot of people, I think you are not an exception, like food, the sense of food. And this importance of feeling or senses give us pleasure from the environment. We like the environment because we can feel the environment. Sometimes patients who are immobilized and extensively bandaged after trauma, sometimes after burns, they can experience visual, auditory, and tactile hallucination, and sometimes morbid fear or panic when these patients are isolated from the sense, when they can't feel anything, can't see, can't feel. And sometimes patients connected to life support equipment in the reanimation department, sometimes they become delirious. Saying in short, I'd like to tell you very important information, that sensory input is vital to the integrity of the personality and intellectual function. So we need to feel in order to be socialized and to be rational and intellectual person. Therefore, in this section and the next lecture, we will consider the sense organs, and we will start, as you see here, with the organ of vision. This is the most, organ of vision, or eye, is the most important sense organ for human. because people receive about 90% of all information from the environment by organ of vision. So it is the most important sense organ in our body. We today, before discuss the anatomy of the eye, I would like to say you at the beginning of this lecture few words about the general properties of all sensitive organs and tell you something about analyze. Let's get started. I'd like to start by defining the concept of sensation. As you see here, this is definition of sensation. It is the conscious or subconscious perception of changes in the external environment or within our body. In other words, in the internal environment of our body. As you see, any sensation are created by sensory receptors and formed in our brain. So, sensation is a function of our brain, not receptors are accepted in this process. But finally, all sensation are formed by our brain. Sense organs or another, this is synonym, analyzer. This word was take in our vocabulary by Ivan Petrovich Pavlov. You see picture of this famous rather great Russian physiologist Ivan Petrovich Pavlov. It is lived in mostly in the end of 19th century and in the beginning of the 20th century. Pavlov defined the analyzer as a complex of anatomical structure receiving irritation from the external environment and from the internal organs as well. Those structure transform signals in nerve impulses, and they conduct nerve impulses to the brain and form sensation there. As you see below, the analyze structure is, there are three analyze structure. First of all, that is receptor, number one, the first part of the analyzer. The second part is conductive pathway, which provides the transport and conducting nerve impulses to the nerve center, and the brain center is the third part of analyzer called sometimes projection center. So at least every analyzer has got three part: receptor as a cells which receives irritation and transform it in the nerve impulses. This is number first part. Then second part, this is a conductive system, some conductive tracts or pathways, and the third part of analyzer is brain center, where we form feeling or sensation. Thanks to our scientist Ivan Pavlov. The next slide show you different types of senses. By the distribution of the receptors in our body, we distinguish two types of sensation. That is the general senses and special senses. What is the difference between them? General sensitivity is divided into, as you see, in somatic sensitivity and visceral sensitivity or visceral sense. Somatic sensitivity is information that we receive from the integument of the body and skeletal muscles. It means that is the information from our skin and muscles. What kind of information we get from the skin and muscles? That is the touch, temperature, the feeling of cold and heat, pain. It is necessary, and you know when you hurt yourself, you feel pain. And proprioceptive feeling. That is the proprioceptive. That is receptor that get information from the tendons, muscles, and joint capsules. They give us the information about pressure, stretch, and muscle tone. All of the somatic receptors are relatively simple in structure. Sometimes they look like a bare dendrite of neurons, nothing more. But sometimes it is a special cellular structure we called capsules or bodies, small bodies. They provide receiving the irritation and transform all of the receptor transform irritation to the nerve impulse. Visceral sense is information about the state of internal organs. The receptors of internal organs detect also heat, cold, and that is the temperature receptors. They are possible. They can get pain information. We know where sometimes when we have pain in stomach, we know that it is a stomach pain, but not is the liver pain, for example. They registrate stretch and pressure in internal organ as well by moving of the organ. And it is very important function. They receive information about chemical composition of our internal environment. This receptor in the internal organ or visceral receptor can also analyze many stimuli that we don't perceive consciously, such as the blood pressure and blood chemistry. So we don't know, nobody knows their blood pressure without manometer. So, but we also don't know the chemistry of our blood chemistry. We don't know concentration of carbon dioxide or oxygen in our blood. But actually, there are some special chemical receptors that though they get information about internal environment. And regulate blood pressure and breathing and etc. A lot of functions are regulated by this, by giving this information and transforming this information to the brain. The special senses, you see all of these senses in the right part of the table, and there are only 5 special senses, not 6, because sometimes we have 6, but that is not real special sense. That is intuition. That is possible to predict future. I'm joking. So special senses are smell, taste, vision, hearing, and equilibrium. And we will talk today about vision. Next lecture we will talk about hearing and equilibrium, but smell and taste we studied when we get a lecture about cranial nerves. And I'd like to demonstrate you, because we started our lecture with the organ of visus, and I'd like to show you the scheme of visual analyzer. As you see here, visual analyzer consists of there are 3 parts in visual analyzer. The first is peripheral department with receptors. That is eye, or eyeball, in other words. The second part of visual analyzers, this is a visual pathway. As you see here, the first part, number 1, that is the eye. Then visual pathways consist of optic nerve, optic chiasm, that is here, that is crossing of optic nerves. Then information go on the optic tract and come into the subcortical centers, 2 centers, that is the superior colliculus and the posterior nucleus of the thalamus. And the last part of the pathway is the optical radiation. That is the fibers between thalamus and cortical zone. So, and the third part of the visual analyzers called visual projection center. Sometimes we call that primary center. It is located in the occipital lobe of the brain. Here, this surface of the cortex in the brain, cortical brain, occipital cortical part of the brain of the telencephalon is the visual primary center in our body. And here you see classification of the organ of vision, and as you see, our eye has 2 parts, that is eyeball and accessory visual structures that help eyeball to function. Eyeball consists of eyeball layers. There are 3 layers: external fibrous layers, intermediate vascular layers, and inner layers called retina. Eyeball, this nucleus, is the second part of the eyeball. It consists of the lens, vitreous body, and some humor or the liquid in our eyeball. So there are 3 parts of the eyeball layers as well as 3 parts of eyeball nucleus. What about accessory visual structures? They have a lot of structure, but we could divide all structures, all accessorial structures, into 3 apparatus. That is a protective apparatus, which protects our eye, a lacrimal apparatus, which produces the tear, and locomotive apparatus, which move the eyes inside of the orbit. If you see another more detailed classification of the organ of vision, you see that eyeball layers consist of the fibrous layers, that is the sclera and cornea, 2 layers. Vascular layers has 3 structure: choroid, ciliary body, and iris. And internal layer or inner layer we call retina. Take a look what structure, what anatomical structure are part of the protective apparatus. That is the fascial sheath of the eyeball, fascial sheath. That is the retroball bar, fat, orbital septum. This is membrane between the orbita and eyeball. Conjunctiva. This is the mucosal layer covering the eyeball and eyelids. Eyelids is also 2 eyelids. We have 2 eyelids, superior and inferior. That is the protective apparatus. Eyelashes, I hope you guess, this is the hair which we see on the eyelids. And eyebrow we are located on the skin of the frontal area of our face. All of this structure are protective structure in accessory visual structure of the eye. And lacrimal apparatus, there are 3 main parts of the lacrimal apparatus. That is, of course, the first is the gland, lacrimal gland. Actually, telling true, there are not only lacrimal gland. There are a lot of small lacrimal gland producing the tear. Lacrimal pathway, which transports the tear to the lacrimal canicule and to the lacrimal sac. All of these structure are the parts of the lacrimal apparatus. And locomotive apparatus, that is the muscles which move the eyeball. I hope now it's clear the classification of the eyeball. That is the anatomical classification, but I'd like to tell you about functional classification of the eye. as you see here, that is there are four systems in human eyes, not only on human but also in the animal human. It is the same. It has the same structure. And first functional system is the shaping system, which forms the eyeball. Actually, this is the fibrous layer of eyeball. The second system is optical system. There are a lot of structure in our eyeball which provides the optical function. First of all, it's cornea. That's a part of fibrous layer. Then liquid or aqueous humor, lens, vitreous body, iris, ciliary muscle. All of this structure provides a passage and retraction and focusing of the light rays. The third system in functional classification of the eye, that is the receptor systems. Only retina is a part of these systems, and it perceives light, form, and encodes nerve impulse and transmits that to the optical nerves. And the last system we called trophic system. That is the vascular layer which provides nutrition because it consists of blood vessels and give all of anatomical structure oxygen and take off carbohydrate gas, and also give all of the structure nutritions like proteins, fat, and everything they need. Here I'd like to overview basic anatomical structure and see them in the sagittal section of the orbit. Here is sagittal section of the orbit, and you see here this is the eyeball. It consists of three layers, external layers, fibrous layers, then brown layer here. This is the vascular layers, and inner layer is retina. All of these are layers of the eyeball. Then I'd like to demonstrate eyeball nucleus. Of course, that is the lens. Then you see large structure named vitreal body, and also you see two cameras, anterior and posterior, which is filled by the aqueous humor. All of this internal structure located in the centrum of eyeball called eyeball nucleus. And then I'd like to demonstrate additional accessory visual structure that help which help eyeball to move, to protect them, etc. First of all, this is the eyelids you see here, and upper eyelids and lower eyelids. They have special hard connective tissue inside. We call it tarsal cartilago. Sometimes you maybe have heard about this tarsal structure, tarsal cartilages in the eyelids. And also you see the muscles which are part of the eyelid. This is the muscles we studied in the myology, and it calls musculus orbicularis oculi, orbicular muscle of the eye, of the orbit. And you see also protective structure called eyelashes, as well as eyebrow, that is the hair. Eyelashes and eyebrow are hairs. Then you see muscles, external eyeball muscles. They move, turn around, turn up and turn down our eyeball. They connected with the external layers of the eyeball and move it. Then you see this protective structure called conjunctiva or bulbar conjunctiva, because this is the mucosal layer covering the eyeball and the eyelids from the posterior, from its posterior surface. And also you see yellow tissue. This is the fat, orbital fat, which its role is like pillow, and it protect dislocation of the eyeball, and that is like some structure maybe protect the eyeball from the dislocation. And now it's time to study eyeball in details. Here you see picture of the eyeball in horizontal section, and I can add that eyeball lies within the orbit of the skull, along with the extraocular muscles. You see here muscles, two muscles connecting to the eyeball, and also you see here cranial nerves. This one, it is also located in the orbit, and we call this nerve optic nerves. I hope you remember that is the second pair of cranial nerves. The eyeball, human eyeball, is smaller than a ping pong ball. I hope you play ping pong or table tennis, and actually the size of our eyeball looks like a ping pong ball, and it weighs approximately about 8 grams. So this is not very heavy structure, and the size is 24 millimeters in diameter. So if we measure the diameter of the eyeball, it will be about 1 inch. Of eyeball total surface area, only the anterior 1/6 is exposed. It means we can see only a small part of our eyeball, and that is the external part. But remainder is recessed and protected by the orbit. I mean, this one is internal part of the eyeball. And as I said, the mass of orbital fat provides padding and insulation for the eye. The wall of eye, I mean, if we take in consideration and study the wall of the eye, we see that it is made up of 3 distinct layers. Here, the outer fibrous layers, called fibrous layers, then intermediate vascular layers, and yellow one is the inner layers. The eyeball is hollow, and the inferior is divided into cavities. Sorry, not inferior, anteriorly. The large posterior cavity here also called the vitreous chamber, contains the gelatinous vitreous body. It looks like a gel. And the smaller and anterior cavity here has 2 chambers. That is anterior chamber between the cornea and iris. They are layers limited anterior chamber, and also posterior chamber here. This is a space between iris from the anterior side, and that is limited by lens from the posterior side. As you see, anterior chamber is deeper and larger than posterior one. The shape of a human eyeball is stabilized by vitreous body. This structure stabilizes the eyeball, but the most strong layers of the eyeball is fibrous layers called sclera. Eyeball layers, anatomically, the wall of the eyeball consists of 3 layers. Let me repeat it again. The external is sclera and cornea. They are parts of fibrous layers. The second, here you see it's in brown color. This is the choroid or vascular layers. Posterior part is choroid, but anteriorly it is iris and ciliar body. All of this structure are parts of vascular layer. And in the end, the most internal layer, this is the retina, which is sensitive. Mostly part of the retina is a sensitive layer which consists visual receptors. And next table, I hope it helps you to understand, to see the classification of the eyeball and summarize all of the structure and understand their function. Here you see function of each layer as well as the function of different parts of the eyeball nucleus, and let us also show you can see this table in details, and I hope it will be helpful for us, for you. Then we see a fibrous layer and called sclera. The outer fibrous layers, tunica fibrosa in Latin, is divided in 2 regions. The posterior region called sclera and the anterior one called cornea. Here you see sclera. It is a white layers which is visible. We see this in the external part of the eyeball, and the cornea is the transparent layers. It is also visible part of the fibrous layers of the eyeball, and we called it sclera. As you see, we now are talking about sclera. The thickness is not the most thickness structure, only from 1 to 0.5 millimeters, but it provides us very important function. It forms the eyeball and protects it. Here I'd like to demonstrate you the changes we can see on the sclera. In a young person it is white and consists no blood vessels and no some inclusions, but in age-related person, in seniors, the sclera turns yellow and accumulates pigments, and it is visible here. This is not white, but it is yellow because of this person is old person, unfortunately. So it is easy to evaluate the age of people. Just take a look on the sclera. Cornea is another anterior part of fibrous layers, and it is located in the anterior part of eyeball, and is its thickness in the center here is about 0.6 millimeter, but on the periphery it is it has more size. Its thickness is about 1 millimeter. Usually cornea is very spherical object, very spherical structure, and the radius of curvature is about 7.5 millimeters. What is the function of cornea? The cornea has the appearance of an hourglass, and it provides conduction and refraction of lights, and it also protects the internal structures of the eyeball. The cornea is rich in nerve endings, but normal cornea has no blood vessels. And of course the most important function is protection of the eyeball from mechanical damage, from toxic damage. It also prevents the lens from the light, especially ultraviolet lights, and it cuts off light rays with a wavelength of 295 up from 295 to the 390 nanometers. Because of the cornea has no blood vessels, it gets nutrition and oxygen by vessels of lymph. They are located around of the cornea, and fluid in the anterior chamber inside of, and of course the tear give some nutrition to the cornea. Actually, we have only three organs in our body where which don't contain blood vessels. There are no blood vessels in cornea, in lens, and in the vitreal body. All of these anatomical structure are located in eyeball. Here you see defect of the cornea. This is a small defect called foreign body. we call here foreign body. I don't know the special term used in your country, but this is an artificial object come into the cornea and damaged it. So this is a reason to come to the ophthalmologist and ask him or her to help, because sometimes that is danger for the eye, and such damages need to be consulted by doctors. And another picture show you the diseases called wall eye or clouding of the cornea. Usually cornea has to be transparent and spherically and specularity, but if it is not transparent, then you see such as blots or spots, white spots on the cornea, and that is a bad situation, because it prevents of the normal vision. Another features of the cornea you see here, and it is interesting for in the point of view ophthalmologist, because this it is unique structure which has high regeneration, high permeability for liquid and gaseous substances, and that is also very good innervated. This is usually when we touch the cornea, we just feel only pain, nothing more, because there are no special receptors except pain receptor, pain nerves, dendrites that provide us the feeling of the pain by touching. And the spherical shape of the cornea is necessary to get normal vision. Here you see normal vision when the cornea is spherical, but when it gets cylindrical shape, we call this astigmatism. What does it mean? Astigmatism is inability to simultaneously focus light rays that enter the eye on different planes. Focusing on vertical lines, for example, vertical lines here, such as the column here, may cause horizontal lines, such as these lines, and caused by a deviation in the shape of a cornea, so that it is shaped like the back of a spoon. I mean, this is a cylindrical form. It looks like a back of the spoon, rather than like part of sphere, and it is a result of the astigmatism. People see objects not clearly, and special lenses which are cylindrical and in glasses, because we wear glasses. People wear glasses when they have astigmatism, and such cylindrical lenses reflect light more in one plane than in another plane. That is interesting part of medicine, and usually ophthalmologist can help people with astigmatism. The second intermediate layer, we call vascular layer, consists of, it is middle layer. We call it in Latin tunica vasculosa. Is also has the name the uvea, because this name uvea was given to the vascular layer because it resembles a peeled grape in fresh dissection. You see here, it is usually it is looks like a peeled grape. So the name uvea come from this reason. The vascular layers of eyeball consist of three regions: the choroid, the first one, the second one, ciliary body, and the anterior, the third part, called iris. See here you see iris. Iris divides the front of the eyeball into two chambers, anterior and posterior. It works as an adjustable diaphragm that controls the diameter of the pupil. Here, this gap, this opening called pupil, this foramen in the iris called pupil, and iris has two pigmented layers. One layer is a posterior pigment epithelium that blocks straight light from reaching the retina, so that is also protecting layers. Iris is protecting layers, and the other epithelium is the anterior border layer of the iris, which contains pigmented cells called chromatophores. High concentration of melanin, this is a pigment, in chromatophores gives the iris a black or brown or hazel color. If the melanin is scanty, light reflects from the posterior pigment epithelium and gives the iris a blue-green or gray color. So, depend on the concentration of melanin in the iris, we can see different color of our eyes. Here you see different colors of the iris, and this picture, I mean the iris structure of the color and topography of iris elements is unique, and in other words, the iris has a very individual color and fiber structures. This is used for personal identification of people, and along with the usual passport control, in some places there is a checkpoint that determines the identity of a person by the iris of eyes. So not only fingerprint, but also iris is the organ or the part of our body which is very, very special and individualized. Here you see different color. When a lot of melanin, the color is dark brown or sometimes dark or sometimes green, but when there is no melanin or small amount of melanin, you see blue eyes and gray eyes as well. It is interesting that inside of iris, we could find two different musculate fibers or muscles. Here you see two muscles in the iris, and they when they contract, they change the pupil. The diameter of the pupil is controlled by the concentric circles. Here you see these circles, concentric circles of smooth muscle cells around the pupil, and these muscles called constrictor of the iris. But also we could find the pupillary dilatator consists of the spoke-like arrangement of contractile processes that extend from the cells of the posterior pigment epithelium, and this pupillary dilatator makes pupil large or wide, and pupillary sphincter produce iris construction and increases the pupil. How it works visible on this picture. Pupil constriction is by circular muscles, and that is a parasympathetic or autonomic system, parasympathetic part of autonomic system. This is a normal position, normal size, and here you see pupil dilates with radial muscles, and they are innervated by sympathetic part of autonomic systems. Such pupil we have in dim light, but when we are on the sunshine, the pupil constrictors make pupil narrow, and when it is very, very narrow, we call this meiosis in Latin. It means that the pupil is small, but when it is large, we call this midrias, and it is a normal size. Sometimes doctors use special medicine, drop in eyes, and make pupil keep pupil dilated, just to see inside of our eyes and examine some internal structure of the eyes. It is interesting video I'd like to demonstrate you that show polychoria, very rare symptom of the eye when people have multiple pupils, not one but multiple, and that is the name of this anomalies called polychorea. It means two, sometimes three pupil instead of one. Ciliary body is the second part of the vascular layers, and it is located posteriorly to iris, and this part of vascular layers is thick and grades into the ciliary body. This is the anterior part of the vascular layer, ciliary body, which is divided into the anterior folds, which consist of processes and ciliary folds. This internal or anterior part of the ciliary body called ciliary pars plicata, what means folded parts. But posterior flat parts called ciliary body pars plana. Here is pars plana and here is pars plicata. What is the function of pars plicata? It produces intraocular fluid, but the posterior pars or pars plana, pars plicata, it is of course Latin name. In other words, pars flat or pars folded in English, and the folded posterior part of the ciliary body change the curvature of the lens. It calls accommodation because it has special connection, special special cords that fixed on the lens and change its curvature. Inside of ciliary body we could find special smooth muscle of the ciliary body. Is called the ciliary muscles. Ciliary muscles. That is the ciliary margin of this section, and with its complex arrangement of longitudinal, oblique and circularly oriented smooth muscle fibers, the ciliary muscles produces a narrowing or sphincter action of the ring-like ciliary body. It means these muscles just pull on the zonular fibers or lens ligament and make when it construct it maybe two way. When lens becomes flat, or another more typical, when some part of ciliary muscles contract, it move the ciliary body anteriorly, and the lens becomes curved. So it change the shape of lens and give us possibility to see far and to see close, to be able to see objects located far from us and located close to us. As I said, construction or relaxation of ciliary muscles changed the tightness of the zonular fibers. Here you see these fibers of the lens, and this is like a film, like cord, change the lens shape. And this mechanism, this function adapts us for vision near or far, adapting us for near or far vision. And as I said, it is called accommodation. What does it mean? Accommodation is the ability of the eye to focus the image of objects on the retina and with different distances. Here you see flat lens, we can see far, but here you see curved lens and we can see close to eyes. Next part of vascular layer is called choroid. The choroid is the posterior portion of the vascular tunic. It is a highly vascular, deeply pigmented layer of tissue between the sclera and retina. You can nicely see here all of this structure, and that is the choroid. Choroid consists of 3 layers. This is large vessels, medium vessels, and the most intermediate part consists of capillaries. We call them choriocapillaries. Of course, as all layers consist of the blood vessels, choroid function is supply retina and another layers with the blood, with the nutritions. The choroid layers, choroid also contains melanocytes. This is special cells. Those produce the pigment melanin, which causes this layer to appear dark brown in color. Melanin, as well as in retina, in the iris, melanin in the choroid absorbs straight light rays, which prevents reflection and scattering of light within the eyeball. As a result, the image cast on the retina by the cornea and lens remains sharp and clear. Some people called albinos, they are white, they have white color of skin, has lack of melanin in all parts of the body, including the eyes. Albinos often needs to wear sunglasses even indoors, because even moderately bright light is perceived as a bright glare dual light scattering. Inner layer of eyeball called retina. That is the third layer. And retina lines the posterior 3/4 of the eyeball and is the beginning of the visual pathway. In accordance with the function of the retina is divided into two parts. The visual part of retina, this is this part of retina, it is yellow, contains sensitive receptors called rods and cones, but anterior part here, where retina covers ciliary body and iris and even lens, that this part is blind part of the retina, and the border between blind and visual and sensitive, in other words, part called ora serrata or judged edge. The retina consists of a pigmented layer and a neural layer. The pigmented layer, you see it here, is a sheet of melanin containing epithelial cells between the choroid and the neural part of the retina. The melanin in the pigmented layer of the retina, as in the choroid, also helps to absorb straight light rays. The neural layer or sensory layer, that is here, is a multi-layers outgrowth of the brain that processes visual data extensively before sending nerve impulses into axons that form the optic nerve. And this neural layer consists of different neural cells. You see these cells here. Histologically, we divided retina into 10 layers, but it is not very important for anatomy. But when you study histology, you will have to know all of these layers of the retina. You see that the external layers of the retina consist of different visual receptors called cone and rods. If we take a look in the scanning microscope, electronic microscope, you see rods rods like intestinal villi, but they are yellow, and green cells we called cone. This is the scheme of cone, and this is the scheme and draft of the rods. There are only two different cells we call visual receptors. As you see here, rods don't detect different color of light. These receptors are very light sensitive and enable us to see in dimly light rooms at twilight or in a pale moonlight. I am talking about now. I'm talking about rods now. Another cells called cone detect different colors, and therefore they provide us with a color vision. There are three types of cones, and their stimulation in various combinations provide the detection of different colors. It's visible here that counts S, M, and L, and they combine together provide us color vision. Thanks to the cones, we can see color, but cones give us sharper, clearer images, but they require more intense light than rods. So combination of cones and rods provide different visual information. Approximately 125 million rods form a broad band around the periphery of the retina. As you move toward the center of the retina, the density of rods gradually decreased. In contrast, most of the roughly 6 million cones, you can just compare 125 million rods, but only 6 million cones. And all cones are concentrated in the area where a visual image arrives after it passes through the cornea and lens. This region, known as the macula, has no rods, and the highest concentration of cones occurs at the center of the macula, an area called the fovea centralis, or simply the fovea. And it provides, fovea centralis is the site of the sharpest color vision. When you look directly at the object, its image falls on the portion of the retina in the fovea centralis. And anatomy of the retina can be viewed with an ophthalmoscopy. This is the instrumental examination when instrument that shines light into the eye and allows an observer to peer through the pupil. It provides a magnified image of the retina and its blood vessels as well as the optic nerve. This is a picture doctors can see during ophthalmoscopy. And this is a scheme. You see the optic nerve. It's a place where form the second pair of cranial nerve called optic nerve. But this is the yellow part. We called it macula lutea, and this is the this is a place where a lot of cones concentrated. That is the best visual area. When we try to see something clear, we try to project this object into on the macula lutea. The surface of the retina is the only place in the body where blood vessels can be viewed directly and examined for pathological changes, such as those that occur with hypertension, diabetes, meltus, cataracts, and age-related macular diseases. So doctors sometimes recommend aged people to be examined by ophthalmoscopy and change their blood vessels. This is another clinical examination, which is modern examination, called high resolution tomography of the retina. Here you see retina, 10 different layers of the retina, and visible the central part of the retina called fovea or macula lutea, yellow spot or yellow macula, and it is very useful for diagnosis some of the retinal diseases. Eyeball nucleus. Eyeball nucleus, that is three part of nucleus. That is a lens. Of course, it is the most important part of nucleus. Then vitreous body and two chambers: anterior chambers and posterior chambers. All of these chambers are filled with the aqueous humor, which is a liquid. This is some kind of biological liquid. And you see this of three parts are presented in the lower part of the slide. Anterior chamber of the eyeball is between cornea and between iris, and it consists aqueous humor. Posterior chamber is a narrow gap-like structure between the lens and between the iris. And as you see, the aqueous humor produced by ciliary process, it's produced in posterior chamber. Then during accommodation, when lens is contracted, the fluid come into the anterior chamber and then go to the blood vessels, special venous vessels located between cornea and sclera called scleral venous sinus, and it provides evacuation of aqueous humor to the veins of the sclera. This is a very important place when people have problems with this sinus, and when it is not possible to evacuate aqueous humor, the disease is difficult and called glaucoma. Usually people feel very strong pain in eyes, and those people need surgical operation, some medicament, some medicine need to be dropped in the eye, and sometimes these people with glaucoma need to be operated, because if they will be not operated, they may lose the visual potential. They may not be able to see. Lens is a spherical structure, absolutely transparent, and you see here the size of, and it have internal structure called nucleus of lens, and external structure called capsula of lens. As you see here, there are four main functions of lens. It is light transmission, light refraction, and participation and accommodation. It is also, and another function is the protective, because it absorbs some bad lights and don't allow them to come to the retina. And this is the different defects of the visual, which is connected with the lens. The normal vision we call emmetropia, normal vision, when objects project right on the surface of the retina. When object project behind the retina, this pathology called hypertrophy, and people need special spherical lens to correct the vision. But more often we have such problem with the eyes called myopia, when the object project before retina, and people need special convex lens just to correct these defects. Presbyopia, this is another problem. This is age-related problem with eyes, when people can't see close to them, and very often not very, but often people have the white spots, sometimes white surface of the lens, and it is visible like a white pupil, and people can't see, they are not be able to see clearly. Doctors just recommend to evacuate lens and replace it with the artificial lens, like you see here. Vitreous body, this is the biggest part. It has a special central canal, we call it hyaloid canal, and this is just form the shape of eyeball. Sometimes when it deformates by deforming vitreal body, we may be problem and give the different spots in vision. That is all for now, my friend, and as usual, let me thank you for your attention and ask you to give me questions if you have. So I'm waiting your questions. If there are no questions, wish you to be healthy, especially now when we have increased numbers of COVID infected people. Is really this number of infected people maximal today. So be healthy and study anatomy well. Thank you for your attention. See you next Thursday, and let me say you goodbye.